Hello friends, in this video we are going to talk about a process called photoreactivation. It's a kind of direct reversal method of DNA repair mechanism. As the name implies, the damage to DNA will be reversed to get the intact working DNA molecule back again. First of all, let's try to understand why this photoreactivation process kicks in within the cell. We have a normal DNA molecule in the cell, and when the cell receives ultraviolet radiations, we know it's kinda lethal to a cell in a way that it damages DNA. So with this UV radiations, there is a covalent bond formation between adjacent pyrimidine residues, and in this case we say we have two thymines adjacent to each other, thus giving us a thymine dimer. And with this thymine dimer, there is a loss in symmetry of DNA molecule. Therefore, now we have a damaged DNA within the cell. And it's this photoreactivation reaction which will remove the dimer. Photoreactivation, as the name suggests, is the light-dependent reaction which involves the visible spectrum of light to initiate the reaction. And finally, with this photoreactivation, the thymine dimer will be removed. First of all, let's see at the molecular level where this dimer is getting formed. We see we have two thymine molecules adjacent to each other, like this. And it's here, these two bonds are getting formed, and we call this a thymine dimer. And this needs to be resolved. Now let's see the mechanism of it, how this thymine dimer is getting eliminated with the use of visible spectrum in this photoreactivation reaction. In the photoreactivation reaction, the photolyases are the enzymes which repair the damage caused by ultraviolet radiations. Photolyases are flavoproteins and contain two light harvesting cofactors. One is called FAD, that's flavin adenine dinucleotide, and the other one is called MTHF, that's methynyl tetrahydrofolate. The folate cofactor MTHF is a light harvesting cofactor and absorbs visible light from the spectrum, preferentially from the violet blue end of the spectrum. Once MTHF gets excited, it transduces energy and electrons towards the FAD, and by accepting the electrons from MTHF, the FAD gets reduced to FADH2. And after that, this FADH2 transfers the high energy electron to the pyrimidine dimer, making a dimer radical. And these pyrimidine radicals are highly unstable and will spontaneously decompose to monomer and monomer radicals anion as shown in the figure. Then finally the dimer radical splits into two pyrimidines, that is the dimer breaks off, and by this way we have the DNA fixed. This photoreactivation occurs in both prokaryotes as well as in eukaryotes. So this is how the DNA gets fixed of dimers formed by the ultraviolet radiations. I hope you like the video. If you like it, give it a thumbs up and make sure to subscribe this channel. Thanks.